Hello scientists! Today we're going to be talking about lesson 3.1, what can a karyotype reveal about an individual with a genetic disease? Please be reminded that this portion of the lesson is supposed to be completed after you've finished the catalyst on page 4, as well as read the background information provided on pages 5 and 6 of the packet. If you've not already completed those items, I do recommend pausing the video now and getting those things done. If you've already finished them, follow along with me on page 7 of the student packet. In this activity, you're taking on the role of a cytogeneticist working in a hospital. Your instructor has emailed you a case study, so go ahead and check your email account and open up that file. Follow the step-by-step procedure which is outlined on the board that your instructor has sent you to arrange the chromosomes into a completed karyotype. Analyze the karyotype and diagnose your patient. This will require a little bit of online research. A warning from us, be careful and use your observational skills as things are not always as simple as they seem. So I've received board H from my instructor and I've pulled it up on the screen so you can see what it looks like. The directions for this activity are located at the bottom of my screen here in the notes box for this Google slide presentation. I'm going to start with step 1. It says read the case study found above. On the cytogenetics report in your packet, record patient information such as the patient ID letter and the reason for the referral. So I can see the patient history is provided in this little folder up in the center of my karyotype board. According to mine, it says that I've been assigned patient H. So on my cytogenetics report for my patient, I'm going to write down letter H. Note that the patients have been given letters to represent them rather than their full names for privacy reasons. My next step is to read that background information under the doctor's progress notes. So according to my sheet, it says that I have a 15-year-old female who's presenting with weak muscle tone, developmental delays, anxiety, and difficulty processing information. An ultrasound of the patient's abdomen revealed that they have ovarian abnormalities. So those are things that I'm going to write down under the why is this patient being referred for karyotyping. So again, from my background information, I see that they have weak muscle tone, developmental delays, difficulty processing information, anxiety, and also ovarian abnormalities. Next, I'm going to be following step 2. To make the process of karyotype assembly less complex, one of the homologous chromosomes is already illustrated on the board. Identify the matching pair. Then click and drag it into place on the board in the proper position. So something that's important for you to know, the chromosomes that are located on the board in this area represent the chromosomes that came from the patient's mother in the egg. The chromosomes that you're going to be dragging and dropping on the left side of the board, those ones came from the patient's father in the sperm. So when the sperm and the egg combined to form the patient, these are the chromosomes that that patient has inside their cells. Something that's really nice is that the chromosomes are actually in order. On the right side of the board, you can see the number of each chromosome underneath. Those do correspond to the orders of the chromosomes over here. So all I need to do is actually drag and drop. And I can see that the banding pattern matches identically here. I'm going to continue dragging and dropping until I notice either some sort of issue with the banding pattern or maybe an extra or a missing chromosome. So far, things seem to be pretty normal. So follow along with me through the process. I'm now on chromosome 6. Nothing seems to be abnormal there. So just a reminder from last class, the first 22 chromosome pairs for my patient that I'm putting on the board right now, those are known as autosomes. Those are all the chromosomes that include lots of different information for all of the things that are important for building my patient, like hair color, eye color, all sorts of different traits and characteristics. The last chromosome on the board that you'll notice in the bottom right corner, those represent the patient's sex chromosomes. So something that's important to remember from the background information, if I have two identical sex chromosomes, they're typically larger as well, those represent a patient who is female because they've received two X chromosomes. On the other hand, if I have two mismatching chromosomes, that probably represents an X and a Y chromosome, which would mean that my patient is a male. Now, I should have two matching X chromosomes, two large X chromosomes in this region right here. If my patient is female, again, I'll have one large and one small one if my patient's male. But as I've put all these chromosomes on the board, I'm noticing something really interesting here. It appears that I actually have 3 of these X chromosomes as opposed to 2. I looked at all the other chromosomes, nothing seems to be abnormal there, so it's probably this extra X chromosome that's related to my patient's symptoms. So that's something that I'm going to want to keep in mind as I'm completing the rest of this cytogenetics report. So let's go back to the report now that I've noticed something interesting. So that means I'm on step 3. I'm analyzing the karyotype for chromosome anomalies, paying particular attention to chromosome number and structure. So for me, it's the chromosome number that seems to be off here. The banding pattern, these lines that I'm seeing on all my chromosomes, they seem to match up normally. There doesn't seem to be any error there. But what's strange is that I have an extra X chromosome, an extra sex chromosome in particular. So I'm going to go ahead and write down my chromosomal findings on my cytogenetics report. So now that I've looked at the bottom right corner, I can tell that my patient's female, or at least they seem to be biologically female, though something's a little unusual about the sex chromosomes here. Do be reminded, if you have 2 X chromosomes, that means your patient's female. If you have an X and a Y chromosome, so 2 mismatching chromosomes here, that means that you have a male. Remember that the sex chromosomes are the only ones where you might not have a perfectly matching pair, and that's if you have a male as opposed to a female for your patient. So now I'm going to fill in this observation. It says compare your patient's karyotype to a normal one. The normal, again, quote unquote normal, karyotypes are posted under announcements on Canvas. You'll see a normal, again in parentheses, a normal biological male karyotype and a normal biological female karyotype that you can compare to your patients. And I can see here the abnormality in mine is the fact that I don't have 2 sex chromosomes, I have 3. So that's what I'm going to write down here. It appears my patient has 3 sex chromosomes rather than 2. In particular, they have 3 X chromosomes. I don't have a Y chromosome. I have 3 X chromosomes. So down in my chromosomal findings, I'm going to put an X for trisomy. The definitions here were provided in that background information. So if you're having difficulty for this chromosomal findings box, I do recommend looking at page 6. This is where you can find all the different changes that you might see in chromosomes or anomalies you might see. On the left side, we have numerical changes. So if you're missing a chromosome or you have extra chromosomes, this is where you want to look. I know that my patient has a trisomy because they have more than 2 chromosomes for a single chromosome. That's different from triploidy, which would mean I have 3 chromosomes for all of my patient's chromosomes. So rather than 46, I'd have 23 times 3 rather than 23 times 2. So I don't have triploidy, I have trisomy. Something that's important to know, it's very unusual for humans to have monoploidy or triploidy. Generally speaking, embryos that have just 1 of each chromosome or 3 of each chromosome do not survive. That is something that you'll see in plants, though, if you decide to become a plant biologist. On the other side here, we'll see structural changes. So if you notice something strange going on with the banding pattern, the lines on your chromosomes, you're going to want to look at this structural change side. Maybe you have a deletion, a portion's missing. Maybe you have a translocation where a piece from one chromosome has been mixed up and shared with another chromosome on your board. So again, if you're having trouble with that box, chromosomal findings, look at the background information on page 6 of your packet. So my trisomy is occurring on chromosome. For me, it's not actually a chromosome number since it's not an autosome. It's one of my sex chromosomes. So I'm going to write sex chromosomes here since it's not a number. And if I wanted to be more specific, rather than just writing generic sex chromosome, I could actually put it's the X chromosome that's abnormal. So I'm going to actually use that to fill in my box here. I have 3 X chromosomes. This last box here, the patient diagnosis, that is going to require you doing a little bit of online research. Make sure that you're using the vocabulary from this chromosomal findings box. That's going to help you identify what the disease is easy. So go ahead and open an internet browser. I recommend using Google. And for me, I'm going to use the vocabulary trisomy X. And I'm going to see what ends up happening when I hit search. So trisomy X chromosome. And I can see, looking here, that most of these searches seem to indicate something called triple X syndrome. So I'm going to go ahead and write that down for now. Can always change it later if that doesn't seem to be what my patient has after doing a little more research. My next job is going to be clicking on a source that's reputable. MayoClinic.org, that seems to be okay, but I really like this one here. This one's from the National Institute of Health, and it's got a .gov, so that seems like a very reputable source out of all the ones that I'm seeing appearing on my screen right now. So I'm going to go ahead and click on that and get a little bit more background information. My job's to see if the description of the disease matches what I'm seeing in the karyotype and in the background information. So according to this, triple X syndrome is also known as trisomy X. That sounds like exactly what I came up with for the chromosomal findings. So maybe I'm on the right track here. It's characterized by the presence of an additional X chromosome in each of a female's cells. Although females with this condition may be taller than average, this chromosomal change typically causes no unusual physical features. I'm going to do a little bit more reading here. Ooh, this seems to match my patient a little bit. Triple X syndrome is associated with an increased risk of learning disabilities and delayed development of speech and language skills. Delayed development of motor skills, such as sitting and walking, weak muscle tone, that matches my patient, and behavioral and emotional difficulties are also possible, but these characteristics vary widely among affected girls and women. Seizures or kidney abnormalities occur in about 10% of affected females. Interesting. So I could do a little bit more background information on here, a little bit more reading through the causes and the frequency, maybe the diagnosis of the condition. Those are things that I might want to look at a little bit more for my patient when I have a little bit more time. After I've finished my research and I've made my diagnosis, the last thing for today's lesson is to write the diagnosis for your patient, arguing what disease you believe they have. Make sure that you're supporting your argument with multiple types of evidence, including the patient's symptoms, the abnormalities you noticed in your karyotype board, as well as the online information that you found during your research. As always, if you have questions, please reach out to your instructor. I'm available for office hours as well this week, so take a look at the announcements on Canvas, see what time that they're posted for this week, and come in with some questions. I'm happy to help. Otherwise, enjoy doing your karyotype board. Good luck.